Hello everyone, today we are going to present the article known as Air-induced juxtasynon is antagonized by TGF-beta to shape calf heterogeneity in pancreatic ductal adenocarcinoma. Here this is our outline. Let's get started with the general information about what is the plaque, its molecular and cellular bases, and finally the tumor microenvironment in plaque. Plaque is the fifth leading cause of death in the USA and one of the major causes of the death in worldwide due to its late diagnosis and low response to treatment, including chemotherapy and radiotherapy. Let me briefly mention what is the pancreas and how the plaque is occur. Pancreas consists of several types of cells, including exocrine, which secrete the digestive juice, and endocrine cells, which secrete the hormones to regulate the blood glucose level, and also the epithelial cells, also called ductal cells. And these are highly elastic. They differentiate into each other in response to the autocrine and paracrine signal, and also the injury. And one of the major processes in the pancreas known as ADM, axonal-ductal metaplasia. During this process, axonal cells, or also called exocrine cells, transferentiate into the epithelial cells or ductal cells. And here, most of the plaque cancer cells arising during this process due to the high prone to the cancer mutations. Some of the mutations are well characterized in the plaque. These include the class activation, which is a very early mutation, and inactivation of tumor suppressor genes, including SMAD4 and TP53, about half of the cases, and finally the constitutive activation of NF-κB, about 70% of the cases. As you can see, the molecular and genetic basis of the plaque is well studied, but there's little known about the tumor microenvironment. In every cancer, tumor microenvironment consists of cellular and non-cellular components. Cellular material include immune cells and the CAF cells, which are called cancer-associated fibroblasts. And previously, the same author identified that PSC, which called pancreatic stellate cell, differentiate. Normally, they regulate the ECM production. Stroma differentiate into two subtypes of cancer: CAF inflammatory and the myofibroblastic. Here you can see the markers. And these two CAF cells create a niche for tumor environment to proliferate the cancer cell and the metastasis. Okay, as I mentioned, same author of this article identified that PSC differentiates to ICAF and MyCAF, but the question is that how this differentiation is controlled by the signaling ligand secreted by the tumor. Okay, in the first result, they investigate whether or not NF-κB pathways are important for the CAFs. For this, they use a PPC mouse tumor. PPC is a genetically modified mouse model to study the plaque. And they check the NF-κB ligands and the receptors marker in the tumor, and they found the high expression of both of them in epithelial cell and the CAF cells. Then they check the presence of IL-1R1, which is NF-κB receptors, and by using flow cytometry analysis, and they found most of the receptors localized on the CAF cell, as you can see in the red area. And finally, they do the single cell RNA sequencing of the CAFs and epithelial cell, and they find similar result that the ligands and the receptors are localized on the epithelial cell and the CAFs respectively. As a result, they conclude that CAFs are responding to the NF-κB pathway. Once they determine the importance of NF-κB pathway, then they specifically show that NF-κB signaling induces the ICAF phenotype. To do this, they use the tumor organoid media, and they check the presence of P65, which is the NF-κB subunit. They extract and fractionate the total protein and do the Western blot analysis, and they found, okay, switch, but this one is a P65 band in the ICAF nucleus, which means that there's active signaling in the ICAF cell. And then they validate this result by inhibiting NF-κB pathway by using IKK inhibitors. And when they inhibit, they see the significant reduction in all ICAF markers compared to the conditioned media in the black box, and then they inhibit in the gray box. As a result, they show that NF-κB signal is important for the ICAF transformation. Now, my friend Batın will continue.
Hello everyone, I'm Batın and I'm with you with second, third and fourth result. And as my friend Deniz mentioned before, NF-kappa-B is important for ICAF phenotype formation, and they look for if there is IL-1 alpha is present and it is causes the ICAF or MICAF phenotype formation. And they look for and if there is IL-1 alpha, they indicated that there is a tremendous increment of ICAF markers, and also these are the JAK-STAT pathway markers, and this is also autocrine signaling, I will be mentioned later. And there is a decrement of MICAF markers, which is alpha SMA marker at the 2, and CTGF is TGF beta target. And we can see that there is some anti-correlation between JAK-STAT and TGF beta signaling. And here they target the antibody and knock out the IL-1 alpha, respectively B and D, and they found the same result. Directly proportional. There is decrement and decrement here, and they knock out the receptor of the IL-1 alpha, and they found that the same result of all these results, and they concluded that IL-1 signaling is important for ICAF phenotype formation. And they look for the and they transplant the tumor into organoids. And knock out IL-1 alpha, and they also found that IL-1A and CST3 are tremendously decreased, and all the other ICAF markers are decreased. And here they transplant the knockout receptor into mouse, and they found the same result. And they look for the tumor size, and they by using ultrasound, and they found that there is a decrement here. There's they look for the ICAF and MICAF cell, and they count them, and there is a decrement in ICAF again, since they knock out the receptor of IL-1 alpha. And after they, so they wanted to see that if IL-1 is the timing. They wanted to see the timing. So after just the 1 hour of exposure, the IL-1 alpha, they see that there is a really important increment in ICAF markers. It's a qPCR results, by the way. And they look for as I mentioned before. These are the ICAF markers and also responsible for JAK-STAT pathway. So they look for the JAK-STAT members, and there is if there is no IL-1 alpha, the phosphorylated JAK and STAT members are decreased. So and then they knock out the IL-1 alpha instead of inhibiting it. Also they see this same result. And as I mentioned before, this is an autocrine signal, and if they inhibit this signal, they also see the same result with JAK-STAT pathway results. And it is a JAK-STAT pathway members, and there is almost no phosphorylation of these members. And also they concluded that there is an impaired induction of ICAF genes. And in here, they inhibit or knock out the JAK and STAT3. And if they inhibit the JAK, and ICAF markers are decreased. And if they inhibit the JAK again, they see that pancreatic stellata cells are stay in the quiescent state, which is a dormant state. And if they knock out the STAT3, and they see that also there is a decrement in ICAF markers, but significantly increased in CTGF, which is a TGF beta. So there is an indirect proportional with JAK-STAT. So if they knock out the JAK and inhibit the JAK and STAT3 knockout, and they see the also same result. These colors are not great to see here because yes, and there is some dots that are red and it is also more censored, and these are the alpha SMA, which is a MICAP, and PSTAT3 is present in the stroma outside of the tumor, and we see that there is a less alpha SMA PSTAT3. So which they concluded that JAK-STAT signaling is low in MICAPs in mouse and human PDA. And my friend Hüseyin will continue.
Until here, Batın mentioned that JAK-STAT is important in IL-1 induction, and in other three results, I'll mention about TGF-beta signaling, and TGF-beta signaling antagonizes IL-1 induced JAK-STAT signaling and inhibits ICAF phenotype. They showed they used TGF-beta signaling markers to show that these markers are increased, and in figure B they used SMAD4, which is TGF-beta effector, and its presence in nuclear site proves that TGF-beta signaling is active. And then figure C, they used single sequence RNA sequencing to show that TGF-beta markers are more present in MICAPs than ICAPs. In figure D, they used immunofluorescence, and they showed that PSMAD2 and alpha SMAs, which are TGF-beta markers, are more populated in MICAPs. And here we know TGF-beta signaling is active in MICAPs in mouse and human product. Actually, in my results, they used mouse and human samples. In here, they did another experiment in the same figure. They showed that downregulation of ICAF gene expression observed is a consequence of TGF-beta mediated inhibition of JAK-STAT pathway. So these are JAK-STAT ICAF markers, and these are MICAF markers. ICAF markers are decreased when TGF-beta signaling is active. MICAF markers were increased, and Western blot analysis also proved this same thing. In figure I and J, they looked for IL-1 receptor 1 expression, and active TGF-beta signaling pathway reduced the expression of IL-1 receptor 1. In other figure, result 6, they showed that the inhibition of TGF-beta signaling targets MICAFs in vivo, but the cell proliferation didn't increase, so tumor growth was not changed, but the stromal region is increased when TGF-beta receptor is inhibited. They used hematoxylin eosin stain and showed the thing by using this method. And also they used Masson's trichrome assay, and they collective result of B and C showed the quantification of the data. Collagen area was decreased when TGF-beta receptors are inhibited. In figure E, they kindly did the same thing by using immunohistochemistry and showed that MICAF marker alpha SMA was also decreased when TGF-beta receptor is inhibited. In figure 7, they showed that JAK inhibition shifts ICAFs to MICAF phenotype. So tumor growth is decreased here when compared to other result, and the tumor growth is decreased in JAK inhibition, and they did same experiments, hematoxylin eosin assay and Masson's trichrome assay, to show that the quantification is exact opposite than the other result. Also immunohistochemistry results were shown, and here we can see that the KPC tumors are increased when JAK inhibitors are used. Thus we know that JAK inhibition results in ICAF state to MICAF states of cancer-associated fibroblasts. In the last figures, figure 7H and I, they show that they used ICAF marker, LY6C, and they show that proliferation is more in MyCAFs. In summary, we can say that there are two main pathways, which one is IL-1 activates NF-kappa B signaling pathway, and it activates LIF, which is autocrine factor that activates JAK-STAT pathway, and TGF-beta signaling is antagonist of this pathway, and when TGF-beta signaling is active, they inhibit IL-1 receptor and leads to formation of MyCAFs. Thank you for listening. Do you have any questions?
Hazırlık aşaması. Elinize sağlık.
I have a question.
Okay, you have a lot of questions. Let's start from there.
In the qPCR result, in the box plot graph, there are discontinued boxes.
First
I don't,
figure.
Tamam, ben çıkayım.
there are lots.
There's lots of
Yeah.
there.
I don't
In
know
here,
these
yes.
illustrations, what does it mean?
Since, as you can see, there is some increment about 0.5, and there is 70. So there is a huge, so they cannot fit the graph, so they use that.
Okay, thank you.
That's how you do it when you have a lot of like changes.
As I can see in this article, 3D environment is important, and they used different models in organoids, mouse and human, you
Yes.
say. I wonder if they indicated the differences between these models in the environment of the disease, phenotype of the disease.
Yes. Most of them, they use the KPC mouse, the genetically modified mouse, and some of them in the Batın GÜLEÇ figure, they use post-mortem samples from the correct cases. But in figure 1,
Result bir mi?
Here they use organoid media, tumor-induced organoid media, and it didn't show, but the supplementary data, they checked the validity of these organoids by checking the ICAF and MyCAF phenotype in the introduction slide. By checking this, the markers, and they said that their model reliable, they recapitulate the disease development organoids. So they get the same result.
So they didn't pick one, probably, but they just tried to
And they
merge the
didn't
results.
highlight any differences between them.
I think they got coherent results, right?
Yes.
They were
Yes,
very
correlation.
Can you put slide 8 number
8.
8? You said that they did the host transplantation, but I couldn't understand why. What's the difference that?
In here, thank you for your question. In here, in normal organoid, they transplant the tumor, and in the other one, the host has IL-1R1 knockout and has normal organoids.
Okay, that is the healthy one, and this is the already cancerous.
No, they actually try to check the which cell secrete and which cell receive the signal. So they inhibit the tumor which secrete IL-1 alpha, and then in second they inhibit the IL-1 receptor in the host which is stromal cell. So they conclude this result.
So they wanted to see is it because of the cough or because of the environment?
Because of the signal,
Knockout or the-
yes,
yeah.
IL-1 alpha and IL-1 NF-kappa signaling pathway, not an environment. Actually, it's both of them.
Mhm.
It controls both of them.
They
Tumor
are-
and microenvironment.
Yeah, I couldn't understand
Mhm.
any, but I understand. Okay, thank you. But I have another question. Is it metastatic or are they inject?
No, it's not metastatic. Just
It's not metastatic.
Yes, it's not.
Okay.
They didn't mention about anything about this.
Are the mouse models are xenografts?
Xenograph.
did they transplant human
Transplantation
cancer
where? How
cells
did they
into mice?
transplant it?
What is the type of the cell?
The primary tumor or cell lines?
They use the primary tumor for the postmortem samples they take from patients. And for KPC, they take from models. And finally, they take the.
I'm not talking about the human samples. I'm talking about the mouse samples. Are they xenografts of human pancreatic cancer cells in mice?
Yes, yes.
Or are they pancreatic cancer cells of the mice?
In mice, right? In mice, they take the primary tumor from mice.
Because PSCs are pancreatic stellate cells, so they must be from mouse, right?
Yes.
So probably they just modulated the receptor expression, right? They did a knockout and then they transplanted
Yes.
and followed how they behave
Yes.
in vivo, right? So, well, thanks guys. So I have a question. So you have, picked a very nice example of how two pathways crosstalk, right? So JAK-STAT pathways controlling TGF-beta signaling, so they have an antagonistic effect. So if you were to just summarize, I just have a methodic question. So the experiments or the perspective of, let's say, the methodic perspective in vitro and in vivo, what kind of experiments did they do in summary?
They check most of them the time the mRNA and protein like
Yeah.
qPCR and Western blot. When
In
they do this
vitro, right?
Yes, in vitro.
In the co-culture and so on.
And they also do flow cytometry analysis in the vitro and in KPC mouse model. And they also use the immunofluorescence microscopy to observe the tumor microenvironments in the postmortem samples.
Okay, and so they didn't only check the inducers of JAK-STAT pathway, but they also checked some targets, right? Targets of the pathway. Because,
Yes.
When they inhibit, they observe the change in the mRNA level.
So these are like the target genes that are
Yes.
regulated by JAK-STAT pathway, some of them, right? So these are TGF-beta, but in the previous ones, I think you have also shown. Where was it? Or maybe it was later. So
The
this
CTGF is a TGF
TGF-beta
target.
target, okay. Okay. Some other questions, right?